Hello friends and welcome to my channel Sequential Biology. Now we are going to start a new series, that is Developmental Biology series. And in this series sequentially we will discuss about structure of gamete, that is we will discuss about structure of sperm and ovum, and also the process of gametogenesis, that means oogenesis and spermatogenesis. And after then we will discuss about fertilization and then cleavage, blastulation, gastrulation, placentation. So different field of developmental biology will be discussed in this series. And today we will be talking about basic concept of developmental biology, and we will discuss the differences in between embryology and the developmental biology. And also we will build few concepts on model organism in developmental biology and the developmental event, that is the fertilization, cleavage, gastrulation, organogenesis. So this concept, you should have clear concept about this developmental event, and we will discuss elaborately later. But here just we will get basic concept about this event, and at last we will discuss the differences in between embryo and fetus. First, we will see the relationship in between embryology and the developmental biology, or the differences in between embryology and developmental biology. This branch of biology is known as embryology, which deals with the study of development of an individual before birth. But in developmental biology, embryology and the genetics are collectively combined. And here in developmental biology is a broad range of biology in which the scientists try to find out the phylogenetic relationship in between different organisms by studying their developmental period. Or simply we can say, when embryology is combined with genetics and the molecular biology, then this branch of biology will be known as developmental biology. Then embryology is the study of development of an individual before birth, and Aristotle is regarded as father of embryology. This is very, very important, and I have highlighted important portion. Developmental biology, by combining molecular genetics with classical embryology, a new branch of biology was originated, and this is known as developmental biology. Then the developmental biology is the study of transformation of a fertilized egg into an adult. Developmental biology reflects the phylogenetic relationship among species. And we know there are present 1.7 million species in Earth. But if we study their developmental process, we can find many similarity of developmental process in different species. And we can easily distinguish which species are closely related and which are distantly related. And with the help of developmental biology, we can find out the phylogenetic relationship among different species. And the biologists have noted the common features of early stages of development among species. And with the help of these common features, we can find out which species are closely related and which are distantly related. Even animals that display widely differing body plan share the many basic mechanisms of development and often use a common set of regulatory genes. And here we can see a very important regulatory gene which is known as the Tin-Man gene. And this Tin-Man gene is responsible for position of heart in different organisms. And here you can see the human and the Drosophila melanogaster, although they are distantly related. Human is chordate and Drosophila melanogaster is non-chordate. But also the position of their heart is determined by a specific set of gene which is known as Tin-Man gene. And this is known as developmental biology, in which the genetics also studied with embryology. This is known as developmental biology. The gene that specifies heart location in a human embryo has a close counterpart with a nearly identical function in Drosophila melanogaster, and the researchers identified that the gene and named this gene as the Tin-Man gene, and the embryo in which this gene determine the position of heart in both human and fruit fly. And the defective of this Tin-Man gene causes the lacking of heart, right? Then this Tin-Man gene are present in both human embryo and the Drosophila melanogaster, and they maintain the positioning of heart in these two distantly related group. And now we will discuss about model organism in developmental biology. Drosophila melanogaster is a very important model organism. In Drosophila melanogaster, axis formation have been studied in Drosophila melanogaster, how anterior and the posterior axis are developed in Drosophila melanogaster, and the dorsal ventral axis, different type of genes have been studied in Drosophila melanogaster. So this Drosophila melanogaster is a very important model organism in developmental biology because it has a short life cycle and the mutant can be easily studied. So Drosophila melanogaster is a model organism. The sea urchin, in sea urchin, the scientists have studied fertilization. Fertilization have been studied in sea urchin, and so sea urchin is a model organism. And in frog and chick, usually cleavage, blastulation and the glastulation have been studied in frog and chick. Nematoda or the Serenohebditis elegans is also an important model organism in developmental biology. But here the human is not a model organism. Remember it, human is not a model organism. Then these are important model organism in developmental biology. Now we will see few important developmental event. The first stage is fertilization. When we start our journey, the first stage of our journey is fertilization, and in which the sperm and egg are united to form a diploid zygote. The fusion of the sperm and egg to form the zygote is known as fertilization. And just after fertilization, this zygote undergo the mitosis division. And with the help of this mitotic division, the zygote will produce many cells which are known as blastomere. And remember, the few egg have yolk, and the yolk always hamper the cleavage process. So the cleavage do not occurs in equal rate in total zygote, and the regions where the yolk are lacking, the cleavage rate is very fast, and this will form the micromeres. And the yolk laden portion, usually the cleavage are slow, and this will form the macromere. So the cleavage is nothing but this is a series of cell division that divide the zygote is known as cleavage. And the zygote will be transformed into many cells, and these cells are known as blastomere. And this is rapid division and the lag growth. And usually we know in cell cycle there are present M phase, G1 phase, S phase and the G2 phase. But in case of cleavage, only S phase and M phase are present. No growth phase can be seen during cleavage. And so the cleavage is very rapid division and it has lacked the growth phase. And the generate a hollow ball of cell which is known as blastula. By the process of cleavage, the blastula will be generated and a hollow ball of cell which is known as blastocell. Then the structure in which the blastocell are present is known as blastula. Just upper side of blastocell, the blastomeres are very small and these are known as micromere. And in the lower side of blastocell, usually the blastomere are large and these are known as macromeres. Anyway, the cells which form the blastula are known as blastomere. And after formation of this blastula, the third stage of our developmental period is occurred, and this is known as gastrulation. And during the process of gastrulation, the blastomere undergo morphogenetic movement. We will discuss about different kind of morphogenetic movement: epiboly, emboli, invagination, evolution, involution. So different type of morphogenetic movement are there. Then the gastrulation is the process by which blastula fold on itself and the rearranging into the multilayered structure. And this multilayered structure is known as gastrula. And by the process of morphogenetic movement, the three germ layers are formed. Then the structure which contains three germ layers, that is the ectoderm, mesoderm and the endoderm, are known as gastrula. And the process by which the blastula is transformed into gastrula is known as gastrulation process. And these three germ layers are responsible for formation of different type of organs. And the last stage of our developmental period is the organogenesis, and this is the fourth stage. The basic germ layers undergo the further morphogenetic movement. The blastomere undergo morphogenetic movement and this will produce the gastrula. And in gastrula, the ectoderm, endoderm and the mesoderm are present. And the cells of this germ layer also undergo morphogenetic movement and this will produce different type of organs, and the process by which different type of organ are produced, this process is known as organogenesis process. And here you can see an embryo in which the organogenesis have been started, and this is the heart, this is the limb blood, this is the brain, backbone. So in this embryo the organogenesis have been started, and the cells interact with each other forming the rudiment of organ. This is the rudiment of organ. The limb bud is the rudiment of our hand, and the rudiment of the organ are formed by the process of organogenesis. And the most spectacular event during the process during organogenesis as the neurulation. Neurulation means formation of the rudiment central nervous system. Central nervous system means our brain and the spinal cord, and also the gut formation. The rudiment gut is known as archenteron, and this archenteron is formed during gastrulation. And the differentiation of the mesoderm, and the mesoderm will be differentiated to form the backbone, muscles, kidney and the heart. And you should have clear-cut idea about which organs are developed from which type of germ layer. This will be very, very important. Then these are the basic event which is occurred during the developmental period, that is the fertilization. This is the first stage in which the zygote is formed, and after then the zygote undergoes mitotic division, and this will form a hollow ball of cell, and which is known as blastula. The cells of the blastula undergo morphogenetic movement, and this will form a 3 germ layer containing structure, and this is known as gastrula. And the archenteron or the primitive gut are also formed in gastrula. And after then the cells of this 3 germ layer undergo again morphogenetic movement, and this will form different type of organs or the rudiment of organ, and this process is known as organogenesis. And usually in developmental biology, these events are studied elaborately with the help of different kind of model organism, as an embryo. And now we will discuss the differences in between embryo and fetus, and this is very, very important, and sometimes we become confused about these two terms. And we all know that in case of human, the gestation period is 9 months or 260 days or 38 weeks. And during this time, we spend our life in our mother's womb, and a small cell structure become billion of cells, that is an embryo will be developed. But here we will be talking about the differences in between embryo and fetus. The first 2 months of our developmental period is known as embryo, and during this stage the most spectacular changes occurring during this time, and the unborn baby acquires its main organs and just begin to recognizable as an human. Then what is embryo? In case of human, the first 2 months of our developmental period is known as embryonic period, and the unborn baby acquires its main organs and just begin to recognizable as human. And after then, from the 3rd month up to the birth, we call the developing individual as fetus. And the embryology is the study of formation and the development of embryo and fetus, right? Then this is the basic idea about our developmental biology.